Welcome to the fascinating world of gene editing, where science meets innovation to reshape the future of medicine, agriculture, and beyond. In this video, we'll explore the basics of gene editing, focusing on the revolutionary CRISPR technology. Gene editing allows scientists to make precise changes to DNA, the blueprint of life. This capability opens up endless possibilities, from curing genetic diseases to improving crop yields. As we journey through this topic, we'll uncover how these techniques work, their potential, and the ethical questions they raise. Let's begin by understanding what gene editing truly means and why it's such a game changer. CRISPR stands for clustered, regularly interspaced short palindromic repeats. It's a natural system found in bacteria, which they use to defend against viruses. Scientists have adapted this system as a powerful tool for editing genes. CRISPR works like molecular scissors, allowing researchers to cut DNA at specific locations. This precision makes it possible to remove, add, or alter genetic material with remarkable accuracy. The simplicity and efficiency of CRISPR have made it the most popular gene editing technique today, revolutionizing research in genetics and biotechnology. Understand how CRISPR works. Imagine a pair of scissors guided by a GPS. CRISPR uses a special protein called Cas9, which acts as the scissors, and a guide RNA, which directs Cas9 to the exact spot on the DNA that needs to be edited. Once there, Cas9 makes a cut, and the cell's natural repair mechanisms take over. Scientists can harness these repairs to introduce new genetic material or disable faulty genes. This process is not only efficient but also highly customizable, making CRISPR a versatile tool in the lab. CRISPR is the most well-known. It's not the only gene editing technique available. Earlier methods like zinc finger nucleases ZFNs and transcription activator-like effector nucleases TLENs paved the way for modern gene editing. These methods also allow for targeted changes in DNA, but are generally more complex and less efficient than CRISPR. Each technique has its strengths and weaknesses, and scientists choose the best tool depending on the specific application. Understanding these alternatives helps us appreciate why CRISPR has become the preferred method in recent years. Gene editing holds incredible promise for medicine. With CRISPR, scientists are working on treatments for genetic disorders like sickle cell anemia, cystic fibrosis, and muscular dystrophy. By correcting faulty genes, these therapies could offer lasting cures instead of just managing symptoms. Researchers are also exploring gene editing to fight cancer and viral infections, opening new frontiers in personalized medicine. The potential to transform healthcare is immense, but it also requires careful testing to ensure safety and effectiveness before these treatments become widely available. Beyond medicine, gene editing is revolutionizing agriculture. Scientists use CRISPR to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. This means higher yields and reduced need for chemical pesticides. Gene editing can also improve the nutritional content of food, helping to address malnutrition worldwide. By making precise changes to plant DNA, researchers are creating crops that are better suited to feed a growing global population, all while reducing the environmental impact of farming. Gene editing isn't limited to plants and humans. Scientists are also applying CRISPR to animals, both for research and agriculture. For example, gene editing can help create livestock that are healthier, grow faster, or produce more nutritious products. In research, animal models with specific genetic changes help scientists study diseases and test new treatments. However, editing animal genes raises important ethical and ecological questions, especially regarding animal welfare and the potential impact on ecosystems. One of the most powerful applications of gene editing is the creation of gene drives. Gene drives are genetic systems that increase the likelihood a particular gene will be passed on to future generations. This technology could help control populations of disease-carrying insects like mosquitoes that spread malaria. While gene drives offer exciting possibilities for public health and conservation, they also come with significant risks, such as unintended ecological consequences. Careful research and regulation are essential before gene drives are used in the wild. With great power comes great responsibility. The ability to edit genes raises profound ethical questions. Should we edit the genes of unborn children to prevent disease? What about enhancing traits like intelligence or appearance? There are concerns about unintended consequences, inequality, and the potential for misuse. Many scientists and ethicists argue for strict guidelines and public discussion to ensure gene editing is used responsibly. Balancing innovation with caution is key to making the most of this technology while minimizing risks. To ensure gene editing is used safely and ethically, governments and organizations around the world are developing regulations. These rules cover everything from laboratory research to clinical applications and agricultural use. Oversight helps prevent misuse and ensures that new therapies and products are thoroughly tested before reaching the public. International cooperation is also important, as gene editing can have global impacts. As the technology evolves, so too must the frameworks that govern its use. CRISPR has become an indispensable tool in disease research. By editing genes in cells or animal models, scientists can study how specific mutations cause diseases. This deeper understanding leads to better diagnostics, treatments, and even preventive strategies. For example, researchers use CRISPR to create models of cancer, Alzheimer's, and rare genetic disorders, accelerating the search for cures. The ability to precisely manipulate genes is transforming our approach to understanding and fighting disease. Gene therapy is one of the most exciting applications of gene editing. By correcting or replacing faulty genes, gene therapy offers hope for patients with previously untreatable conditions. Recent breakthroughs using CRISPR have shown promising results in treating blood disorders, blindness, and even some forms of cancer. While challenges remain such as delivery methods and long-term safety, the progress so far suggests a bright future for gene therapy powered by gene editing technology. Editing the genes of human embryos is one of the most controversial aspects of gene editing. While it could prevent inherited diseases before birth, it also raises concerns about designer babies and unforeseen consequences. Some countries have banned this practice, while others allow limited research under strict conditions. The debate continues as scientists, ethicists, and policymakers weigh the potential benefits against the ethical and societal risks. This area remains one of the most closely watched in the field of gene editing. CRISPR is also being used to fight infectious diseases. Scientists are developing CRISPR-based diagnostics that can quickly and accurately detect viruses and bacteria. In addition, gene editing may help create new treatments by targeting the genetic material of pathogens. For example, researchers are exploring ways to use CRISPR to disable the DNA of viruses like HIV, potentially leading to cures. These innovations could transform how we detect and treat infectious diseases in the future. Gene editing is playing a key role in improving food security. By creating crops that are more resilient to climate change, pests, and diseases, scientists are helping ensure a stable food supply for the future. CRISPR allows for faster and more precise breeding compared to traditional methods. This means new crop varieties can be developed and brought to market more quickly, helping farmers adapt to changing conditions and feed a growing population. Gene editing is also being explored for environmental conservation. Scientists are investigating ways to use CRISPR to protect endangered species, control invasive species, and restore damaged ecosystems. For example, gene editing could help revive populations of threatened animals or make plants more resistant to disease. While these applications are still in the early stages, they highlight the potential for gene editing to support biodiversity and environmental health. Public perception plays a crucial role in the development and acceptance of gene editing technologies. While many people are excited about the potential benefits, others have concerns about safety, ethics, and unintended consequences. Education and transparent communication are essential to build trust and ensure that the public can make informed decisions about gene editing. As the technology advances, ongoing dialogue between scientists, policymakers, and the public will shape its future. CRISPR has become a cornerstone of modern biotechnology. Its precision and efficiency have accelerated research and development in fields ranging from pharmaceuticals to industrial enzymes. Companies are using CRISPR to engineer microbes that produce valuable chemicals, biofuels, and even biodegradable plastics. The technology's versatility is driving innovation across multiple industries, making it a powerful tool for solving some of the world's biggest challenges. One of the main challenges in gene editing is delivering the editing tools to the right cells in the body. Scientists are developing various methods, such as viral vectors and nanoparticles, to improve delivery efficiency and safety. The choice of delivery method depends on the target tissue and the specific application. Overcoming these challenges is essential for realizing the full potential of gene editing in medicine and other fields. While CRISPR is highly precise, it's not perfect. Sometimes the editing tools can make changes at unintended locations in the genome, known as off-target effects. These unintended edits can have unpredictable consequences, especially in medical applications. Scientists are working to improve the accuracy of gene editing and develop methods to detect and minimize off-target effects. Ensuring safety is a top priority as gene editing moves closer to clinical use. Rapid development of gene editing technologies has led to complex patent and ownership disputes. Multiple research groups and companies claim rights to key CRISPR inventions, leading to legal battles over who can use and profit from the technology. These disputes can affect access to gene editing tools and slow down research and commercialization. Resolving these issues is important for ensuring that the benefits of gene editing are widely shared. As with many new technologies, cost and accessibility are important considerations for gene editing. While CRISPR has made gene editing more affordable, there are still significant expenses involved in research, development, and clinical applications. Ensuring that these advances are accessible to people around the world, not just in wealthy countries, is a major challenge. Efforts are underway to reduce costs and expand access so that the benefits of gene editing can reach everyone. Fully realize the potential of gene editing, education and training are essential. Scientists, healthcare professionals, and regulators need to understand the technology and its implications. Universities and research institutions are developing new programs to train the next generation of experts in gene editing. Public education is also important, so that people can make informed decisions about the use of these technologies in society. Gene editing is a global endeavor, with scientists and organizations around the world working together to advance the field. International collaboration helps share knowledge, set standards, and address challenges that cross borders, such as disease outbreaks and environmental issues. By working together, the global community can ensure that gene editing is used responsibly and for the benefit of all. The future of CRISPR and gene editing is full of promise. Researchers are constantly improving the technology, making it more precise, efficient, and versatile. New applications are emerging in fields like synthetic biology, regenerative medicine, and even space exploration. As we look ahead, it's clear that gene editing will continue to shape the future of science, medicine, and society in profound ways. Synthetic biology is an exciting field that combines gene editing with engineering principles to design and build new biological systems. Using CRISPR, scientists can create organisms with entirely new functions, such as bacteria that produce medicines or plants that clean up pollution. This field is pushing the boundaries of what's possible in biotechnology and could lead to breakthroughs in health, energy, and the environment. Gene editing is paving the way for personalized medicine, where treatments are tailored to an individual's genetic makeup. By understanding a patient's unique genetic profile, doctors can use CRISPR to develop targeted therapies that are more effective and have fewer side effects. This approach is already showing promise in cancer treatment and rare genetic diseases, marking a shift toward more precise and personalized healthcare. CRISPR is transforming plant breeding by allowing scientists to make targeted changes to plant DNA. This can result in crops with improved yield, nutritional value, and resistance to disease and environmental stress. Unlike traditional genetic modification, gene editing can create changes that are indistinguishable from natural mutations, making it a more acceptable option for some consumers and regulators. Gene editing has the potential to both support and challenge biodiversity. On one hand, it can help protect endangered species and restore ecosystems. On the other, there are concerns about unintended effects on wild populations and genetic diversity. Careful research and monitoring are needed to ensure that gene editing supports rather than harms biodiversity. CRISPR is not just for editing genes, it's also being used to develop rapid and accurate diagnostic tests. These tests can detect genetic mutations, infectious diseases, and even cancer markers with high sensitivity. The speed and accuracy of CRISPR-based diagnostics are making them valuable tools in both clinical and research settings, improving patient care and advancing scientific discovery. Researchers are exploring the use of gene editing to understand and potentially slow the aging process. By studying genes involved in aging and age-related diseases, scientists hope to develop interventions that promote healthy aging and extend lifespan. While this area is still in its early stages, the potential to improve quality of life as we age is an exciting frontier for gene editing research. Microbes are tiny powerhouses that play vital roles in health, industry, and the environment. Gene editing allows scientists to engineer microbes for specific purposes, such as producing medicines, cleaning up pollution, or improving gut health. CRISPR has made it easier and faster to modify microbial genomes, opening up new possibilities in biotechnology and environmental science. Patients with rare genetic diseases, gene editing offers new hope. Many of these conditions have no effective treatments, but CRISPR can target and correct the underlying genetic mutations. Clinical trials are underway for a range of rare diseases, and early results are promising. As research progresses, gene editing could become a lifeline for patients with conditions that were once considered untreatable. Veterinary medicine is also benefiting from gene editing. CRISPR is being used to develop treatments for genetic diseases in pets and livestock, improve animal health, and enhance breeding programs. These advances can lead to healthier animals and more sustainable agriculture, while also raising important ethical and regulatory questions. Scientists are beginning to explore the role of gene editing in understanding and treating mental health conditions. By studying the genetic factors involved in disorders like depression, schizophrenia, and autism, researchers hope to develop new therapies. While this research is still in its infancy, it could eventually lead to more effective and personalized treatments for mental health. Cancer research has been transformed by gene editing. CRISPR allows scientists to identify and target the genetic mutations that drive cancer, leading to new treatments and personalized therapies. Some clinical trials are already testing CRISPR-based approaches to treat various cancers, with encouraging early results. The ability to precisely edit cancer-related genes is opening up new avenues for fighting this complex disease. Gene editing is advancing our understanding of the immune system and enabling new treatments for immune-related diseases. By modifying immune cells, scientists can enhance their ability to fight infections, cancer, and autoimmune disorders. CRISPR is also being used to develop next-generation vaccines and immunotherapies, offering new hope for patients with challenging conditions. Regenerative medicine aims to repair or replace damaged tissues and organs. Gene editing is helping scientists create stem cells and tissues with specific genetic traits, paving the way for personalized transplants and treatments. CRISPR is accelerating progress in this field, bringing us closer to the goal of regenerating damaged body parts and curing degenerative diseases. As humanity looks to explore and settle other planets, gene editing could play a vital role. Scientists are investigating ways to use CRISPR to help plants and microbes survive in harsh space environments, support life support systems, and even protect astronauts from radiation. These innovations could be key to long-term space missions and the future of space colonization. Bioethics is a critical field that examines the moral implications of gene editing. Questions about consent, justice, and the long-term impact of genetic changes are central to the debate. Bioethicists work alongside scientists and policymakers to develop guidelines that ensure gene editing is used in ways that respect human rights and dignity. Ongoing ethical reflection is essential as the technology continues to evolve. Public policy plays a major role in shaping the development and use of gene editing technologies. Policymakers must balance innovation with safety, ethics, and public interest. Effective policies can encourage responsible research, protect public health, and ensure equitable access to new treatments. As gene editing becomes more widespread, ongoing policy development will be crucial to guide its responsible use. With the rise of gene editing comes the need to protect genetic data privacy. Genetic information is highly personal, and its misuse could lead to discrimination or breaches of confidentiality. Laws and regulations are being developed to safeguard genetic data, ensuring that individuals' rights are protected as gene editing technologies become more common. Gene editing has the potential to reshape society in profound ways. From healthcare and agriculture to ethics and law, its impact will be felt across many areas of life. As we navigate this new era, it's important to consider not just the scientific possibilities, but also the social, cultural, and ethical implications. Open dialogue and thoughtful decision-making will help ensure that gene editing benefits everyone. As we look to the future, the road ahead for gene editing is both exciting and challenging. Continued research, responsible innovation, and public engagement will be key to unlocking the full potential of CRISPR and other gene editing technologies. By working together, we can harness these tools to improve health, protect the environment, and create a better future for all. Conclusion: CRISPR and gene editing techniques have ushered in a new era of science and medicine. Their potential to cure diseases, improve food security, and address environmental challenges is truly remarkable. However, with this power comes the responsibility to use it wisely, ethically, and for the benefit of all. As we continue to explore and develop these technologies, let's strive for a future where gene editing is a force for good, shaping a healthier, more sustainable world.